Okay, so have you ever really thought about it? How do scientists find one single gene, just one, hidden inside the billions and billions of letters of our DNA? I mean, it's a huge challenge, right? It's like finding a needle in a haystack if that haystack was the size of a planet. Well, today we're going to dive into a super clever technique that was invented to solve this exact problem.
For decades, really, the main strategy was to basically play genetic detective. The classic approach, was to break a gene on purpose and then just watch, see what happens. You're looking for some kind of noticeable effect, kind of like following footprints to find a suspect. But, and this is the big but, what happens when those footprints just disappear? What do you do when the trail goes completely cold? And here's the thing, for a lot of genes, that trail went cold all the time. The problem was, the genetics rulebook, well, it had some pretty big loopholes. For starters, a lot of genes are redundant. That means if you break one, another gene can just swoop in and take over its job, totally hiding the fact that anything's wrong. Then you've got others that are doing all sorts of things at different stages of development, making it almost impossible to pin down what their one job is. So you could break a gene and nothing, no obvious change. You're just left scratching your head, totally stumped. So, okay, the classical approach was hitting a wall, a real dead end. Geneticists needed a totally new game plan. They realized they had to stop just following the clues and start setting traps. And that brings us to the big aha moment, gene trapping. This is so clever. Instead of just randomly breaking stuff and hoping for the best, this technique is way more sophisticated. Basically, it involves sticking a specially designed piece of DNA, the trap, into the genome at random. Now, this trap has two crucial parts. First, a reporter gene that basically screams, "Hey, I got something." And second, a selectable marker that helps you find that screaming gene later. It's an absolutely brilliant tool for those genetic detectives. Okay, so how do you actually build one of these things? Let's pop the hood and take a look at the Geneticist Toolkit. All right, building the perfect trap comes down to three key decisions. First, you've got to build the gene trap vector. Just think of the vector as the special delivery package that carries the trap. Second, you need an insertion vehicle. That's the delivery truck, if you will. It's how you get that package into the DNA. And finally, number three, you have to pick the right reporter gene. And this is so important because that's the signal that tells you your trap actually worked. And just like any good hunter, a geneticist knows that one size does not fit all. Not all traps are the same. You've got to choose the right strategy for the specific kind of gene you're hunting for. So let's compare the two main strategies, because this is really interesting. Think of a gene's promoter as its on switch, okay? So on the left, you've got an enhancer trap. This trap comes with its own but very weak on switch. It's designed to find other little bits of DNA called enhancers that act like a volume knob, boosting that weak signal. Then on the right, you have the promoter trap. This one is totally different. It has no on switch at all. It's completely silent until it lands right inside a gene and basically hijacks that gene's on switch to get itself heard. So the key takeaway here is it's a trade-off. There's no single perfect trap. The enhancer trap, it's awesome for finding those specific enhancer sequences, but it doesn't really mess up the gene much, and you have to screen a ton of cells to find what you're looking for. The promoter trap, on the other hand, is way more likely to actually break the gene's function, but the chances of it landing in exactly the right spot are pretty slim. So it really is like choosing between casting a really wide, gentle net or trying to hit a tiny target with a very precise spear. Okay, so we've set the traps, the hunt is on. The next big question is, how in the world do you know when you've actually caught something? And this is where that reporter gene we talked about comes into play. It really is the secret weapon here. It acts like a genetic beacon, sending out this bright, unmistakable signal the moment the trap lands inside a gene that's actually turned on. I mean, how cool is this? One of the most famous reporter genes is called GFP, and it originally came from a jellyfish. So when a gene trap carrying GFP actually works, the cell literally starts to glow bright green. You can physically see the discovery happening right under the microscope. It's amazing. And believe it or not, it gets even cooler than that. Another option is the luciferase gene, which, comes from a firefly. With this reporter, a successful catch doesn't just glow under a special light, it makes its own light. The cell literally becomes a tiny biological lamp, putting out a signal that scientists can measure with incredible precision. But seeing that glowing light is really just the first step. The next part is the genetic screen. Scientists use that selectable marker we mentioned to grab only the glowing cells.
And from there, they can do a couple of things. First, they can do an expression screen, which basically tells them where and when that trapped gene is turned on in the body. Then they do a genotypic screen, using powerful tools like PCR to make millions of copies of that bit of trapped DNA. That lets them read the exact genetic code and finally figure out which gene they've caught and give it a name.
So, what does all this mean? What's the bigger picture here for science, for medicine?
Yeah, this quote here really says it all. Searching the unknown with gene trapping. It's not just a cool lab trick. This gives us a systematic way to explore the huge, uncharted territories of the genome. And finding these hidden genes, it isn't just about making a big list of parts. No, it's about understanding the actual instruction manual for life itself. And, more importantly, finding the typos in that manual, the genetic errors that can lead to diseases like cancer or Alzheimer's. This is exactly how we build better models to study these diseases and, hopefully, design smarter ways to treat them.
So ultimately, this technology completely changed the game. It took the search for genes from being a total shot in the dark to being a targeted, efficient expedition right into the code of life. It's a foundational tool that's behind these huge international projects to map out what every single gene does, which, of course, leaves us with one last, kind of mind-blowing question. With these incredibly clever traps in our toolkit, what other secrets are still hiding in our DNA, just waiting to be found, just waiting for that little beacon of light to finally reveal them?